So I have a word to say just before we start, if you do agree. So I will first welcome to all of you to attend this workshop. It's a workshop dedicated to transnational issues in nanomedicine. For those who don't know me, I'm Fabrice Navarro, coordinator of New Deal project. This workshop today is co-organized by New Deal and ETPN teams, and with the support of a sister project, Cupido, Be Smart, and Smart for Primary. I will address a special thanks to Nadine Bresch, Suresh Raman from New Deal, and also the two Alexandre from the ETPN for their great help in organizing this workshop. So just to remind you that the aim of our workshop is to share our experience in developing innovative nanomedicine and preparing their future clinical translation. The program is organized with two panel discussions, one dedicated on a clinical development plan led by Alan Boyd, and the second one on manufacturing consideration, so led by Frank Pavon. We would like this discussion as more interactive as possible. So we will appreciate your active participation and feel free to share your concern, ideas, or questions. I'm also honored to introduce our first speakers to launch our workshop. And I would like to underline their quality as experts in their respective fields. So first, Dr. Ruth Schmid, current chairwoman of the ETPN with a great experience in developing medical technology at SANTEF, a Norway research technology organization. She is an expert on chemistry and on polymer particles, and she is going to present the opportunities and challenges in nanomedicine. This will be followed by a talk given by Professor Alan Boyd, an expert in pharmaceutical medicine and clinical and medical affairs. He was involved in promoting the clinical translation of many innovative therapeutics, and is going to introduce just after the associated challenges and the trends. So Ruth, the floor is yours, let's say. Please, both of you, stick to your allocated time and stay attentive to the chat if you will receive information on the time by Suresh. Okay, so Ruth, I'll let you present your talk.
Okay, thank you very much, Fabrice, and thank you for inviting me to very shortly give you some ideas on the evolution of nanomedicine from the the earlier time, and also some ideas on how it will evolve into the future and which are still the challenges. Just to remind you, nanomedicine has a wide range of applications from diagnostics all the way to regenerative medicine. Nanomedicine is in the interface between biology and physics, and it supports the effect of the biological molecules, both the chemical drugs and the biologics, to get a better effect of those molecules. But also nanomedicine opens for totally new therapeutic tools that are based on physics. This is the Gartner hype cycle for new technologies. This is a method that shows how novel technologies normally develop over time, from an idea to high expectations, then a valley of death in a way, a disillusionment, and then it starts enlightening again to the plateau of productivity. And I would say that nanomedicine now is in the slope of enlightenment. And why is that? It is because we change from a pure academic development phase to a really systematic translational approach. Also, the basic understanding built up of interactions between nanomaterials and biology over a long time, they show now results. So we understand it much better, and we can design the nanopharmaceuticals and the nanomedicines in a better way. And I will show you three success stories that really show the paradigm shift in nanomedicine. The first one is Vixeos. It shows a first clinical approved nanoparticle that delivers a synergistic combination of three drugs. The molar ratio is very important in this application, and with the liposomes, we can deliver this drug at the right place to the right time in the correct molar ratio. So this paves the way for new combination nanoparticle formulations. In 2018, Onpattro was approved, and this is the first clinically approved RNA therapy for IV delivery of an RNA, and it's a nanoparticle product. But it is also the first approved RNA product in general. So this demonstrates how nanomedicine and nanoparticles can be used to enable the delivery of highly challenging therapeutics to humans. The last example is Hensifi, which is a totally novel product. It shows a unique mechanism of action, and it represents the next generation of therapeutics that can provide a therapeutic benefit in a complementary and possibly synergistic way to standard therapy. In this case, we talk about enhancing external radiotherapy with a physical mode of action. A few translational facts for nanomedicine. Currently, more than 50 nanomedicine formulations are in the market. More than 400 nanomedicine formulations are currently in clinical trials. And if you look at the percentages of the clinical indications, we can see that cancer still is the most is the highest share, but we can also see that the percentage of cancer clinical trials compared to other clinical indications is decreasing. So this is very good news. Nanomedicine enters into novel clinical areas more and more. If we look at the nanoparticle classes that are in clinical trials, the liposomes are clearly the biggest share, both in cancer trials and in non-cancer trials still. But there is one group of nanoparticles, the lipid-based nanoparticles, that are quite a big share for non-cancer trials, and this is connected to the fact that approximately 8% of clinical trials are in fact RNA nanoparticles. Unfortunately, I don't have numbers for other biologics. All these numbers and these graphs and more information you can find in the opinion paper of ETPN in the Journal of Controlled Release. Let me have a look into the future R&D environment as I see it. I think in the future it will be very important that key enabling technologies are used and developed in synergy, because together and in synergy, the R&D evolution will happen. Let me take one example. If we look at the change from small synthetic drugs to biologics, this change is only possible if you have a nanoformulation for the biologics, because they have to be delivered inside the cells. But the development of nanopharmaceuticals is only possible if we have smart production facilities and processes. And to design the nanopharmaceuticals, we need to understand the disease biology, and we need the biomarkers. So all those key enabling technologies, they are connected, and together it will then be possible to address unmet healthcare needs with innovative nanotechnology-based solutions for the benefit of the patients. There are still gaps. And these gaps have to be addressed and will be addressed during the workshop here, which is a very good idea. There is still a lack in access to relevant preclinical characterization and safety assessment protocols and facilities. And there is a lack in access to scale-up and GMP production lines. There is an uncertainty and fragmentation in the regulatory framework. And there is a lack in access to clinical trials that are adapted to and relevant for nanomedicines. There is also a lack in education, in business management, in academia. And last but not least, it is really important to have a mix of public and private funding to succeed with nanomedicine translation. Thank you very much for your attention. And I'm looking forward to all the discussions in this workshop.
Thank you very much, Ruth. I will let Alan to complete the introduction by with his expertise.
Thanks, Fabrice and Ruth. Can I just check that you can see my slides?
Yes, we do.
Yeah. Okay. Well, thank you very much.
Perfect.
Just to continue on from what Ruth has said, my expertise, as Fabrice introduced me, is in translational medicine and then translating those ideas into medicines to be used as prescription products, and hopefully to get them registered as prescription products for available for patients. Now, in approaching the development of any medicine, put at a very simple level, we need to demonstrate 4 things to the regulatory authorities. We have to be able to manufacture a product, and this product must have, and we must guarantee consistent quality, so that this batch that we might make now is being the same as the next batch that we make is the same as the batch that we've made before. And this is required clearly in required quantities to suit and satisfy the patients that may need it, be it a rare disease or a huge indication like asthma or hypertension. So it's all about manufacturing quality that we have to think about, and think about that early on. The next thing we have to demonstrate is that the product is safe and well tolerated in the patients that are to be treated. Thirdly, we have to demonstrate that the product is effective and is producing the therapeutic effect that's required. And then finally, we have to put all this together and do a benefit risk assessment to make sure that in the indication you're treating, that the benefits that are brought are much better than the risks. And of course, depending upon what treating, so for instance, if we're treating pancreatic cancer and late stage pancreatic cancer, the patients will take many more risks and be prepared to suffer a lot more adverse events than, say, if we're developing a new headache treatment for a 3-year-old. But no matter what the product is, be it a chemical entity, be it a biological, a gene therapy, and a nanomedicine, it's these 4 things that we have to concentrate on and make sure that we deliver. And it's no different for nanomedicines. Now, on this slide, what I've done is outline schematically the process of what we need to do to take a research idea all the way through to regulatory approval. And on my next slide, I'll just talk briefly about some of the problems and issues that we have along the way, particularly in relation to nanoparticles. And during this whole workshop, we'll then be devoting ourselves to talking about some of these issues and how we can overcome them. As you'll probably be aware, typically this whole process takes anywhere between 10 and 12 years. And costs at the moment, from information and data that's been issued, anywhere between £1.5 to £2 billion for a product for a major indication. So the first activity, obviously, is all the preclinical and research activities, and that will then lead on as well to thinking about how the product's manufactured, and the manufacturing process clearly will carry on with the improvements and how it's all made all the way through development. Once we've decided to take a product into development, we then have to do, the toxicology and biodistribution studies and the preclinical assessments in animals, and then move on to clinical studies, first with the first in human studies, first in patient studies, and on to the phase 3 confirmatory studies, where we'll definitely show the product is safe and effective in the indication. And hopefully, if all that comes together, then we'll have a regulatory filing and regulatory approval. But alongside that, we must take the government agencies with us. And there has to be really an early engagement with the regulators, and then a continuing dialogue along the way to make sure we're still on track, and hoping to get their support eventually for a regulatory approval. As to where the issues might be, As I've outlined, they come really in 5 main areas. Sorry, and I've just realized there's a spelling mistake, there's an error on there. That word should be toxicology, preclinical toxicology, not manufacturing. So even in the early days in the preclinical research, developing nanoparticles, we have to get proof of concept. But being a nanoparticle, it isn't sufficient. So for instance, if we're using, say, RNA, siRNA, as the active compound and wanting to produce the therapeutic effect, even at the preclinical research stage, we have to take into account we're going to be putting that structure, that RNA, inside a nanoparticle. So it's important that we do some of the preclinical assessments with the nanoparticle we're thinking about that will carry the RNA, for instance, into the cells for us and get going. In terms of manufacturing, some of the barriers there, again, we not only have to make the active ingredient, but we also have to make the nanoparticle and bring those two together in a combination product, because that's going to be vital. And so that all has to be worked out. And clearly later on today, we do have a session on large-scale manufacturing, which will cover many of these issues. The next issue is the preclinical toxicology, and we'll talk about that more in the clinical development plan session. Because again, we not only have to assess the active ingredient that we're trying to produce the pharmacological effects with, but also it's going to be administered as a nanoparticle. So that brings different issues to the toxicology assessments and where we'll be going. Finally, there's the clinical and then the regulatory activities. Now, performing the clinical studies is no different with nanoparticles than we would do with other clinical studies and with other products that we're developing. Through that program, we have to establish the initial safety and efficacy in volunteers, if that's the route we're taking, or more often than not, with the biologicals that we're developing, we'll go straight to patients. And show that the drug has initial efficacy and safety, and then continue it with the confirmatory phase 3 studies. And so those will set their own challenges, just like any clinical development would do in terms of endpoints, the numbers of patients, the inclusion-exclusion criteria. And then finally, there's the regulatory activities. And again, it does get a bit more complicated because we are dealing with not only the active ingredient, but the fact it is a nanoparticle and how that's produced, how it's delivered, and all the assessments we need to take part of. So really, that's just a sort of some of the highlights, I think, where the potential barriers will be. And of course, during the rest of the day, the rest of this afternoon, we're going to be addressing many of these issues in a lot more detail. So I'll finish there, and I'll hand back now to Fabrice to take